To begin today's event, we would like to recognize and acknowledge the traditional land and territory on which today's event is taking place. It's important to make note of the relational and contextual quality of this protocol. We would like to begin this by acknowledging that the land on which we gather is the traditional territory of the Anishinaabe Mississauga, adjacent to the Mississaugas of Scugog Island First Nation and the territory covered by the Williams Treaty. This place is and will continue to be home to Indigenous peoples. Let us move forward together with kindness and respect. I'd like to take a moment to thank everyone for joining our virtual presentation today. Our speaker this afternoon is Dr. Eva Sapi, who will be presenting on the potential connection of Borrelia infection and breast cancer. Dr. Sapi received her PhD degree in genetics from the University of Eötvös, Lőrincz, Budapest, Hungary. She's a professor at the University of New Haven, Connecticut, where she teaches undergraduate and graduate level biology courses and carries out Lyme disease research with her students. To date, over 100 undergraduate and graduate students have received training in Lyme disease and breast cancer-related research. Her studies investigated the different forms of Borrelia burgdorferi to better understand how Borrelia can hide from the immune system as well as antimicrobial therapies. Her recent studies examine the pathogen-host relationship between Borrelia and mammalian cells, particularly with normal mammary epithelial and breast cancer cells. We'll have Dr. Sapi present, and then we'll open for questions from the audience. You can ask questions by entering your questions into the chat box or raising your hand using the icon. Please help us welcome Dr. Sapi to the podium.
Thank you, Atina, for a welcoming introduction. And let me share my screen. Share screen. Okay, and let me start with the first slide, of course. And let me put it. Okay, and here I'm gonna just move this here so we can see the slides very well. So again, thank you very much for inviting me for these presentations. As Atina mentioned, I work at University of New Haven and I'm teaching and I'm doing research on Lyme disease and recently breast cancer related research in the last almost 20 years. So I always put up this slide because I want to remind everybody that Lyme disease was discovered back in 1976 when 36 children came down with some unusual juvenile arthritis, juvenile issues. And at this point, ER researchers visited a little town called Orland, Connecticut, and started to investigate what could be behind it. And it took a couple of years when 1982, Willy Bordefer actually discovered a spirochete, which might be a causative agent, which later was confirmed that indeed it is a causative agent for Lyme disease. So when we talk about Borrelia, the causative agent, everybody think about the spirochete form, yes? And I find this video on YouTube, practically what this video shows, and we also confirmed in our laboratory that the spirochete form very easily turns into other forms when the spirochete feels threatened. For example, here we're adding penicillin antibiotics into the culture media, and you will see within seconds, the spirochete started to change morphology and become, we call it now as round bodies. Actually, it's a very interesting slow motion presentation of this whole transformation. As you see, it shows like a little bumps occurs, and just a second later, this whole spirochete turns into these round bodies. So, so at this point, when we started to study Borrelia and antibiotic sensitivity, we were very interested in different morphological form. On those green panel here, you can see this typical spirochetal form. That's what you see under the microscope if you look at the culture. But as you see, those round bodies, they're everywhere. At least 10% of the cultures always contains round bodies. Think about round bodies as like a backup. If something happens, we have a pool of cells which is very, very resistant to anything. And in the last 10 years, other forms also discovered, for example, the famous persister forms, which is forms after antibiotic treatment. And all groups discovered the biofilm forms, which is, we believe, that the most resistant forms among those forms. In 2011, we had a big studies when we looked at all the different forms and exposed it to standard antibiotics like doxycycline, tigacycline, amoxicillin, metronidazole, and tinidazole. And we wanted to see which form are the most resistant. And this is the big table. I think it's better if I show you the actual image. So among all those forms, we found that this, at this point, we call it aggregates because we have now proved it is really biofilms. So we found that the aggregates forms are the most resistant forms. Since then, we learned this is in a culture. If we put these forms onto like a collagen layer, that even becomes even more resistant. So on this picture, you could see after treatment, we visualize the dead cells with red stain, the live cells with green stains. As you see, it doesn't matter which antibiotic we used, we can see lots of green stains after treatment. So you'll probably ask, what is biofilm? Biofilm is usually a collection of microorganisms. Could be just bacteria, that could be bacteria, yeast, or even protozoa. I can give you an example. Your teeth, for example, could have those plaque, which is practically a slime forms on the surfaces. Also on your water, on your shower, when you don't clean it for maybe a couple of days, you start to feel the slimy surfaces. And this slimy secretion is a very typical manifestation of biofilm because it's a protective layer. This is like a polysaccharide which protects the biofilm. So no antibiotics or no immune system really can reach biofilms. Clay Costerton actually believed that most of the organisms, they do live in biofilm forms. So unfortunately, they can form almost anywhere when water is present, but also in the catheters, kitchen counters, showers, water pipes, as I mentioned. It can damage industrial equipment. It can contaminate food. It can cause energy loss for inefficient energy transfer. But the most important for us, unfortunately, they are really resistant to antibiotics. So after 2011, we spent a good 10 years to identify the different characteristics of this biofilm form. And one of the major characteristics is that is a structural rearrangement happening during development. So we use something called atomic force microscopy, which can have a very high resolution images of during the biofilm development. And if you look at the left panel, you see spirochetes on panel A, which is started to form biofilm. Look at how nicely aligned the spirochete and creating almost this like crochet-like structure. And this crochet-like structure, if you wait, this is 21 days on panel F. If you wait like 3 weeks, you can see this biofilm and you can see these holes, and you could probably ask, what is this holes in the biofilm? Just think about it. If you cover this surface with this protective layer, you still have to bring in some nutrition, and you can still have to expel waste. So these holes are channels, which is again very, very a classical manifestation of a biofilm. This is a high magnification of these channels. And also some protrusions we found on a biofilm, which we still try to understand what is exactly for. But what you see here is really definitely a very important structural changes during biofilm formation. Of course, you ask me, so what kind of protection this biofilm can have? And and this is back in 2012 when we found one of the major one called alginate, which is very typical of can be found on other biofilms. This is just a picture from the paper when we showed that alginate indeed present in this Borrelia biofilm. And here is just a spiral heater from you see it's hardly any staining. The last row is just some unspecific staining for the reviewer to accept or concept. So we published after this paper several other papers. For example, we identified that this biofilm forms in, for example, 37 degrees Celsius better than lower temperature, which is interesting because that's exactly the mammalian temperature we have. We also found that other species, not just Borrelia burgdorferi, but the European species also can form biofilms. So we tried to do a lot of different studies to better understand it. Of course, when I say better understand, we wanted to better understand to able to eliminate it. Now, in this presentation, I'm not going to present most of our antimicrobial studies. I would like to focus in this presentation a little bit more on biofilm and what happens in a biofilm in different tissues. So back in 2012, when we published the first paper, there's a lot of people said it is only going to happen in the lab, in the test tube. Who knows what you guys doing, but it's not real. So it was very important for us to able to prove this concept by using actual tissues. I was lucky enough to get some tissues from Dr. Zager from Austria, from some non-Borrelia infected skin tissues. And I also got some tissues from a very famous case, autopsy tissues from Dr. Ligner and Dr. Goldman, who fortunately and unfortunately had this patient who received long-term antibiotic treatment, and despite of that, she died. I'm gonna talk more about this in a later presentation. So again, it was very, very important for us to able to prove that this Borrelia biofilm form indeed can be found in human tissues. It's not just in a lab artifact. So as I mentioned, I got some skin tissues called Borrelia lymphocytomas, which was already used for that other study. It was already proven that is Borrelia in it. So we were absolutely confident at this point that, you know, if it's a biofilm form, that would be the best tissues. And I always show this because this was the very, very first proof that biofilm indeed exists in tissues. If you see this, so this from the skin tissues, you can see the Borrelia, you can see the beautiful spiral heater form, but also this aggregate showed up, which is stained with alginate, remember, for the biofilm marker. So that was the very, very first proof that indeed maybe biofilm can exist in the tissues. This is just on the same paper. We use silver staining, classical silver staining, to further prove that it's really Borrelia, next to some immunostaining. And of course, we try to present as many pictures as we could to show that indeed it's not just a rare occurrence, but it really can happen in this Borrelia infected tissues using, again, immunostaining. Even we provided some, we call the inside to hybridization staining, just to be sure that, you know, what we're seeing here is real. I remember I talked about the atomic force microscopy. We love this little microscope. We put the tissues under this microscope, and this is the image when what we're looking for in this image. So this is Borrelia in these tissues with alginate staining. This is how the tissue look like. And we put it under this microscope, and what we were looking for, channels. Remember those channels? It was very, very important for us to find those channels. And with atomic force microscope, we indeed find the channels. So we know that we're dealing with something similar to what we see in a lab. Of course, at this point, we asked, is other pathogen can find in this tissue? Because it was always a question. So when you have a tissue, and there's a lot of data coming out from some metagenomic analysis, can we, okay, we find one pathogen, but how about some other pathogens in the same tissue? And which one is really most important, you know, in terms of the disease? So for that one, I got lucky because I had some help from Perkin-Elmer Genomic Center. This is the folks who actually worked previously in back in when we discovered the whole genome sequencing. So they were very knowledgeable, and I asked them if it can help us to use their tools and see if, other than Borrelia, what other species can be present in these tissues. So they did all these bioinformatics tools, and they found something called chlamydia species, which I got very interested because just a previous conference, I heard chlamydia is very frequently found next to Borrelia. So of course we started to study it, and we used the same tissues. Of course, here's again Borrelia. Here's chlamydia, looks like a very similar staining pattern. Alginate in this case is blue because I needed the red channel for chlamydia. And at this point, we use something called confocal microscopy, and you're going to see a lot of confocal microscopy images. So think about confocal microscopy, that it can see everything in 3 dimensions, which is very important for a biofilm, of course. So we put this image under this confocal microscopy, and as you see, Borrelia shows up in a green channel. Chlamydia is inside of the Borrelia biofilms, almost like hiding there. And if you look at alginate, the blue channel is sort of covering the whole thing. So it was one of the proof that this Borrelia and this Borrelia biofilm is not just we put a monospecies, so not just one species lives in, but it looks like it's inviting friends. Of course, now at this point get even more interested. So we looked at some other skin tissues which we were able to access. In that case, I know you heard about the Morgellon disease. That's again a skin condition which we believe that caused by Borrelia. So we were questioning, so in this skin condition, what we can find. And we did a very similar analysis I just told you, and we find Helicobacter. Yes, the Helicobacter H. pylori, which you probably heard about also, stomach issues. But if you Google it and try to find some paper, you could see Helicobacter pylori can be anywhere. It could be in your brain, it could be in your skin. As a skin condition caused by H. pylori is quite common. So we practically repeated the same study. We did the previous skin tissues. And again, this is different. So this is corresponding to 3 patient sample. As you see, we indeed find very similarly, Helicobacter looks like very similar pattern to Borrelia. Alginate again is blue. We had some non-specific staining just to be sure what we're seeing is not just some kind of artifact. And I'm sure you heard about that, in Alzheimer's disease, there is these amyloid plaques. When I started to work with skin tissues, I started to understand this amyloid plaque can happen in any kind of organs. It's not just the brain who's sensitive to it. So, and especially I was reading a lot of paper that can happen in skins. So I was at this point very, very interested whether I can see some amyloid changes. So what we did, we stained the same tissues with thioflavin. That's one of the staining pathologists use. Looks like it's staining. For amyloid, I had 2 different antibody. Very interestingly, only one antibody stained, this in blue, which means is probably a specific kind of amyloid. But that also was good to see that not any kind of antibody can stain these tissues. So we did believe there's some kind of amyloid changing happening in these biofilms. Of course, when we talk about amyloid, you probably heard about phospho-tau and how important also those phospho-tau changes, the fiber changing. So we stay in the same tissues for phospho-tau, and we found very, very similar patterns. All right, so again, we use confocal microscopy. Remember, chlamydia was inside the biofilm. In the case of Helicobacter, it looks like it's sort of bending on a closer to the surface on this biofilm. So it's definitely looks like every pathogen has the certain preference how they cooperate or involved in this biofilm formation. Yeah, sorry. Of course, at this point, we got excited because, biofilms are known that a lot of different tissue can have biofilm formation. So we tried to understand maybe we should look at some other tissues. And of course, the brain was one of the other target, especially what we see with amyloid changes. And again, we got lucky again. We were able to get another famous case. This case was a Lyme disease and Alzheimer patient. So we did very similar studies. We used the amyloid, which I mentioned it worked very well for the skin. As you see here, we found this is for the frontal lobe. We found changes. Same with Borrelia aggregates, also staying with alginate and amyloid marker. The last row is just negative controls. We also able to do some interesting histochemistry staining, and on the histochemistry staining, actually, you can see almost a spiral hit even better than on the immunostaining. And this is just again an insight to hybridization picture to further. No, we have an insight to, but this is the phospho-tau picture again, just to prove further that this amyloid change is indeed happening. All right, so I mentioned about this famous case, the woman who got actually long-term 16 years of history of Lyme. The doctor was Dr. Kenneth Ligner, who is a very passionate doctor, and he really believed that he can save this patient. He tried different antibiotics. I had numerous conversations with Dr. Ligner about this case, and I understood that he really tried everything. Unfortunately, at the end, somehow the insurance company stopped paying for the treatment. She couldn't get the antibiotics, and that's when died very suddenly. The silver lining was of this tragical event that the autopsy tissues was stored in Colombia, and we were able to get a grant from Global Lyme Alliance and able to really analyze this tissue for different markers. And again, this is the case when it's culture, comfort, treatment failure. As you see, is reported back in 1992. That was a very first case when it was a very firm evidence that this patient has Lyme disease, and it doesn't matter what kind of treatment she receives. Doesn't look like that she's able to recover from it. So I'm showing this one. This is not partially unpublished because some pictures in the paper, but that was the very first slide we got from Colombia, and that was a liver slide. And I never forget when we looked at this staining, and I had to be honest, I couldn't believe it. This is a huge biofilm. We never see anything. So we stained with different. This is a polyclonal, monoclonal, just different antibodies, to be sure that what we're seeing is real. As you see, the non-specific didn't stain. Alginate again is blue here, stained very well. This is just you can see the ICS differential interference microscope is showing you, actually, you can even see the biofilm in this one. And this is some H&E, which is a standard pathological staining. So we were shocked by this result. So I sent back this result to Dr. Goldman, and I said, can we get more slides? And he sends more slides from, at this point, he had a heart, kidney, liver, brain. And we started to see this biofilm forms everywhere. And this is just a quantitative analysis. As you see, we could per slide, but we can have as many as 6 biofilm, but most often it's two or 3 biofilms, and they're quite large in size. And the other question was, so we have this biofilm. This patient died with massive inflammation. So we wanted to see if the biofilm actually causing this kind of inflammation. So we looked at for example, CD3, which is infiltrating lymphocytes, one of the biofilm marker. And as you see, kidney and liver, we found the brown staining next to the biofilm. They are very significant lymphocytes there. Again, we use confocal microscopy to see how it looks, under a little bit 3D dimension. There's the heart tissue, and you see the spirochete almost like lining up here. And we have here one, looks like biofilm. Remember the alginate, how do we know? Because alginate is blue at this point, and you see there's very significant alginate staining on these autopsy tissues. Of course, we looked at other other markers. This is not published data. We looked at brain, heart, kidney, liver again. And this is just different inflammatory markers. As you see, some of the markers showed up better than other markers, but we had some inflammatory markers in every tissue. So after we finished this study, we really wanted to explore a little bit the interaction with Borrelia and the tissues. And Borrelia does something. Borrelia creates these outer membrane vesicles. There's a little bubbles coming out from Borrelia. And we wanted to know whether it really has something to do with the inflammatory markers or other markers. So we had this interesting study that that we exposed these outer membrane vesicles. We isolated from Borrelia and exposed it to the neurons. And we find that this somehow affects the superoxide production of these neuron cells, but it was almost like it's down-regulating it, which is interesting because almost like this membrane vesicle is sending us like little primary soldiers and sort of cool down the cells a little bit so Borrelia can come in very easily. I think I have. So the other thing we learned from this study that Borrelia can indeed really infect the cells. So we have this, again, confocal 3D images. I have a video, just 1 second. And it looks like the Borrelia can go right inside in the cells. And I think you can see on this one, so this is the neuron cells. And as you see, Borrelia is right inside. The Borrelia is green here. This is one of the best little video. Let me show it one more time so you could see the cells, the neuron cells. Oh, sorry about that. Let's go back. And neuron cells, and as you see Borrelia green, you can see the spirochete is definitely inside the cells. So as you can imagine that we got really interested in this one. What happens when Borrelia in these mammalian cells? And one of the things happened that it was, I think, is in Warsaw. It was a conference, and I just was having some lunch, and suddenly I had these nurses finding me, and they did not know about my studies, especially they know I'm really looking for Borrelia in a different tissue environment, and they asked me if I ever looked at neoplastic tissues, and I said not yet, but why? And they said because this is unusual precedent that people, especially women, coming down with breast cancer, even some very nasty breast cancer after Lyme disease treatment. So they asked me if I would look into it. Probably they knew my background. Before I worked with University of New Haven, I was actually at Yale, and I was a cancer biologist. So of course, triggered my interest at this point, you could imagine. And I mean, it's not a new thing because we know from 1991 that Borrelia is associated with some B-cell lymphoma. Also, 2008, it was approved that non-Hodgkin lymphoma also has some kind of risk of after Borrelia infection. And Dr. McDonald did find some Borrelia in some glioblastoma tumors. So it wasn't that strange request. So I tried to make some pilot study just to see if we can maybe find Borrelia in these tissues. So fortunately, it's not that difficult to find breast cancer tissues anymore because it's commercially available. It's a little slides which can have as many as 200 200 little biopsy tissues. So you can very quickly stain them for Borrelia and evaluate whether it's present or not. And the result was actually very interesting, because when we purchased this slide, we find significant numbers of invasive breast ductal carcinoma, and some of the lobular carcinomas were indeed positive for Borrelia. We saw a spiroid. I'm going to show you some pictures and biofilms. And of course, we had some controls, some healthy tissues, or some fibroadenomas. They were negative. So probably you asked me how it looks like. So this is, I can show you, because we're not going to use these slides from upcoming paper. We have now, I'm going to mention later, is a very much larger number of tissues we examine. So here is an upper panel. You see Borrelia in breast cancer tissues. You see a very, very classical spiroheter forms. On the right, the E panel, normal tissue, absolutely negative for Borrelia. When you ask me, so about biofilm, we did find some biofilm. I have to be honest, it wasn't as many as we found some the skin or brain tissues, but indeed it was some biofilm was present. And this is, again, I can show you this picture because we're not going to publish it. We have now new pictures, confocal pictures. This is just one of those, what you just seen, cases when we put it under the confocal, and to see better what exactly the pattern. As you see spirohete in rats, and you see, I see some aggregates also. So again, so Borrelia and breast cancer. I know that's all maybe too much to graph quick for the first sight, but according to the American Society, 15 actually now they updated, even 20% of all cancers can have some kind of infectious agents. I mean, this is just an example for the bacterial infectious agents. And as you see from, again, H. pylori in gastric cancer, chlamydia in lung cancer, chlamydia in this lymphoma, mycoplasma, chlamydia in ovarian cancer, and we have evidence that Staphylococcus, E. coli, Bartonella also can be prevalent in breast cancer. There was a very interesting paper published in 2018 when they looked at a lot of bacteria present in different types of breast cancer. And of course, I was very interested to see that they found Borrelia, of course, they found chlamydia, and they find Helicobacter. All those players, you know, we had seen before. Now, saying that, you can see that other bacteria also present. However, the paper specially mentioned that Borrelia presence was associated with poor prognosis. And I had to show you this paper from 1907. That was long time ago, of course. When they looked at spirochetes and cancer in mice, and actually they claimed that that has to be some kind of relationship in breast cancer and this spirochetal organism. So that was the very first evidence more than 100 years ago. And this paper came out last year, and this is, I think, a milestone paper. This is they looked at different microbiota in the breast cancer, and they wanted to see if it contributes to the metastasis. And indeed, they found it. It is a super interesting paper. If you're interested in this topic, please read it, because especially at the end, when they treated the mice with different antibiotics, they found that the metastatic potential of this cancer reduced. So super interesting paper. So first question was, of course, can we see Borrelia inside of breast cancer cells? So we started to culture some breast cancer, especially triple negative breast cancer. You know, that's the worst type of breast cancer. And we started to look into whether Borrelia can invade them. We did publish it, but this is one of the unpublished data. So this is from one of the panels. And as you see, when you say Z-stack is when you look into the tissue, and you can look at it. Oh, sorry about that. You can Z-stack is right here. You can see this better that's really inside. So at this point, we finished the analysis for a couple of different breast cancer cells. These are triple negative breast cancer cells at different time point. As you see, as the time point increases, 3 days later, a lot of spirochete can be present inside the cells. How do we know it's inside the cells? If you look at these red little arrows, you can see that shows that it's inside the cells. This is one of the best images. And, I heard about that, people talk about that biofilm can form inside the cells with the intracellular biofilm, but I have to be honest, I was a little bit, I didn't understand. But when I see this image, look at the middle of this image. I can see sort of like an aggregates forming. I can see a couple of spirochetes start to get together. So maybe this intracellular biofilm is not a strange idea. And I have a video, and the video jumped. So this is the same one as a little bit, but you can see it's definitely inside of the cells. So, okay, so it is there. So what is gonna do? Obviously that was a big question, and the question was, can it be a role in tumorigenic changes in these breast cancer cells? So this paper is published 2021, 2 years ago. And as you see, we used in this paper just 2 cell lines. One on the lower panel, you see the triple negative, and the upper panel, you see a normal epithelial cells. And the first question is, if in fact these cells with Borrelia, can they invade more? Now, when I say invade more, that's again in a lab condition. As you see, MCF10A, which is the normal one, did not change much. 231, which is the triple negative breast cancer cells, here is without infection. It does invade some, is a breast cancer cells. But look what happens when we infect it with Borrelia. It is definitely quadrupled. On the left side, you see different markers, which is people using to affect whether certain tumorigenic changes happening. There's something called the epithelial mesenchymal transition, which is when the stairs start to move. And this is just the markers for it. So we looked at different markers and we saw some changes. Of course, we wanted to see which are the most changes. The most changes we see here, that's called MMP9. What is MMP9? Matrix metalloproteinase. This is the enzyme which the cancer cells secrete and sort of digest the environment so they can move. So that was the most significant data. It did some quantitative analysis, and as you see here, MMP9 was the one of the significant. Not saying that, some other markers also went up, but again, this matrix metalloproteinase was the most important in this experiment. So probably you asked me, so what's happening now? So on this slide, the screening over 400, actually is more like 600 by now. And we co-stain it for different tissue remodeling and of course inflammatory markers. Remember the famous case, we find some inflammatory markers. So we will see if Borrelia induces those increases. The one which actually right now publishing and just right after this talk, I'm gonna upload one of the paper, and next week the other paper gonna go. So we did ask, so what happens? Borrelia infect these cells? What kind of pathways? What kind of genes, proteins gonna start to express in the breast cancer cells to able to make these oncogenic changes? So the one is called RNA-seq, which is the technique to find genes which is upregulated, and the other one called microRNAs analysis. This is a small RNAs, again, very, very important tumor development. So both studies actually concluded, and and a super interesting result. So I'm hoping you're gonna see it very soon. Again, one paper gonna go today, next paper gonna go next week. So hopefully by the summer you can read both papers. There's a tremendous amount of data there, but just to highlight it, we do see genes, which is very, very important. It's not just an inflammation, but cancer development significantly upregulated. Summer project. On the summer project, when we did this analysis, we find a lot of chemo-resistant genes was upregulated. So we have a project right now to look at some whether if we treat the breast cancer cells, some chemotherapeutic agents before and after Borrelia, does it make Borrelia, these cancer cells, even more resistant to those chemotherapeutic agents? I have preliminary data. It looks like it might, but I don't. That's why we're gonna spend the whole summer to be sure what we're seeing is real. Right? Of course, the question will be back to the tissue work. Can we see on those breast cancer tissue? Can we see some other pathogens? Can we see chlamydia, or can we see H. pylori, or maybe Bartonella? Bartonella also was recently suggested to have something to do with breast cancer. So that's other project we're gonna do this summer. And the last project we gonna start this summer. We're going to see other hormone-sensitive cancer like ovarian endometrial cancer. We already have some preliminary data, and looks very, very interesting. So I have a student who's going to spend the whole summer and try to identify whether all those other hormone-sensitive cancers also can be infected by Borrelia. So this is again we're publishing this data. So I'm just gonna quickly show. This is as I mentioned. This is so interesting. What happens after the infection? Left panel, you could see some normal cells. Right panel, you could see the triple negative cells. And as you see, a lot of different pathways are affected after Borrelia infection. Okay. Of course, you asked me, so how about the biofilms? Are you still doing some biofilm research? And actually, yes, we have a project which is almost finished. We were again got lucky. Dr. Jennifer Miller provided us some mouse tissues infected with Borrelia. And actually in that one, we were very interested in the heart tissues, because heart carditis is one of the major symptoms of Lyme disease, and we wanted to better understand that. So in this one, again, we got lucky because she also gave us some tissues which was treated with doxycycline to see what happens if you treat the mice during infection. And what the interesting thing was, it was treated before introduced Borrelia. Again, the studies is almost done. Super interesting. And again, we're hoping that it's gonna get a lot of insight what happens in the heart tissues after Borrelia infection. And of course, we're still searching for antimicrobial agents. Sorry, I didn't have time. I'm looking at time to really explore, the antimicrobial studies. That we studied not just standard antibiotics, but herbal agents. The novel antibiotics is a lot of different things we explored. I have a new student who hopefully he's gonna bring some new energy, some new ideas, because we still need to find some effective and safe treatment to treat Borrelia biofilm, especially in tissues. And here's my summary slide. So again, in the beginning, I mentioned that Borrelia does have a very antibiotic-resistant form. Biofilm is the most resistant one, and we found evidence it can reside in human infected tissues. Biofilm, as you see, I showed you some of those beautiful structural slide, provides a very effective refuge strategy for those from antimicrobial treatments. And it's not just that. People asking me, what do I think about Borrelia and a different infection, especially if we find some other bacteria, maybe even inside the biofilm? My theory at this point that Borrelia making this amazing castle. I call the iron castle, which is a wrong name because Borrelia doesn't use iron, but still looks like an iron castle. And this castle actually can help other bacteria like chlamydia, who does use iron. Maybe that's why chlamydia is in this iron castle, to provide iron to the castle. Or H. pylori, who is obviously another human pathogen, which we still have to learn how to deal with. So they always, even cancer research or even in infectious disease research, they always think about the driver. So who is a driver here who makes this all kind of changes? And people ask me, why don't you study Bartonella? Why not study other? I really believe that Borrelia, this ancient bacteria, is figured out how to protect not that itself, but also protect, making environments from other pathogens, and they can symbiotically can create these extremely resistant forms. We provided evidence that Borrelia can infect various tissues and actually affect cell physiology. And as I mentioned, we have evidence that Borrelia indeed can enhance cancer cell invasion and induce multiple cancer-related pathways. All right, so this is just my last couple slides at the end. This is my team. Next to me is Niraj, Genevieve, Ananya, Miranda, Sambuta, Sahaja, Katie, and Vishwa. Amazing group of people. I cannot be more proud of them. And here's my thank you slide. I always thank the University of New Haven College of Arts and Sciences because they always provided the perfect place for our studies. They built a beautiful lab for us. I couldn't believe it when we moved in. And it's not just that they every time the lab got some kind of criticism, at one point even some request that maybe it should be shut down. University of New Haven College of Arts and Science was always protecting us. Of course, I need to thank all the Lyme disease foundations. They always provided support. And recently for the breast cancer studies, the Pink Clover Foundation and Portman Foundation who are funding this initiative. And you saw state-of-the-art images, and this is thanks for LymeDisease.org, Lyme Disease Association, Schwartz Foundation, Global Lyme Alliance, because they were helping us to buy those high-end microscopes. And I'm pretty sure that will be some questions, but somehow you don't have chance to send those questions. Here's my contact information. Shoot me an email, and we can. I hope I can answer all those questions. Thank you. Thank you.
Thank you, Dr. Sapi, for your wonderful work and years of dedication to this work. We appreciate you sharing it. And now I would like to take some questions, if there are any questions from the participants. Okay, I'll start with one of mine. How do you distinguish between the patient research and studies, similar to those in your presentation, where you were dealing with untreated Lyme models or misdiagnosed Lyme models? And how do you modify the protocols?
Okay, so some of you might know that I also had Lyme disease, and I was dealing with devastating disease. So, to me, it's very close to my heart when I talk almost daily for Lyme disease patients who either misdiagnosed for years or have not received the right treatment. This is why research is so important. I'm hoping, for example, I mentioned this mouse study at the end and treating the mouse with doxycycline and see if doxycycline can indeed prevent those biofilm formation. And I have to tell you, it did not, which is we have to understand this, because I don't know how it's in Canada, but here in US, if you go to your doctor, let's say you even have the bullseye rash, so there is no doubt you have Lyme disease. Your test comes back positive. What you got? Most likely doxycycline for 2 weeks, yes? If you're lucky, you get another 2 weeks. And now we understand this is not the appropriate treatment. Fortunately, we have some Lyme doctors who understand that, and they try different treatment. But Athena, we still have to understand, how to treat this disease. It's not over, unfortunately.
Thank you. Next question: are there presently antibiotics that will penetrate the biofilm?
Okay. The Trojan horse. I'm sure everybody knows the Trojan horse. So how to bring the antibiotic inside this extremely protected structure. So we try different. We have Dr. Horowitz ask us to do the Dobson study. It is you can find it on the internet. That looked like it's some effect. We tried a lot of different combination. It might have some effect. Probably some of the stevia study. Now stevia we believe is the Trojan horse, because this biofilm still has to eat. Yes, this is still living cells in this biofilm. So it has to take up the nutrition. Those channels has to open up. That is one of the things which we would love to see, how this closed channels opens up, and when you send the food there, has to open up. So we believe stevia being you use it on your probably coffee or tea, being these sweet leaves, we believe that it triggers maybe this opening the channels. It gets in, but stevia is used in Japan for centuries as an antimicrobial agent. So again, here's the thing: we found agent which looks like it's working. However, if our studies is mainly done in test tubes, what we learned the last, let's say, 10 years, that the minute Borrelia is embedded in this collagen-rich tissues, and this is an additional protection for them. And let's not forget that Borrelia can go inside the cells. I show you videos of that. We know if the Borrelia is inside the cell, that's an additional layer of protection. So when you say how to penetrate this, I think we have to trick Borrelia biofilm. But again, I'm still struggling how to trick the biofilm when it is so embedded in collagen-rich tissues.
Thank you. Next one, Dr. Sapi, thank you for this wonderful presentation. What is the prognosis for chronic Lyme patients with breast cancer? And would these patients benefit from additional screening with an underlying infection?
Oh, thank you for this question. This is our goal. I think that's if you ask me why we want to rush out these papers, we want to let the physician knows if they have a Lyme disease patients, please screen for not just for breast cancer, because I mentioned we looking at some other cancers, but just be aware that they might be more prone to other cancers. So for breast cancers, yes, definitely some additional monitoring. And prognosis, again, let's hope we can get it early with this additional screening, and let's hope that's not going to causing additional issues for this patient who already devastated by Lyme disease.
Thank you. Where can one locate your antimicrobial research that you referred to in the presentation?
Oh, I think 99% of our paper is free online. So simple Google search, just Google my name, and it's all coming up, and they're free online. And if somehow you don't find one of them, just email me, I'll send it to you.
Thank you. Monolaurin appears to be a very safe and useful product that is antibacterial, antiviral, and so on, and pathogens can't develop resistance to it. Are there any studies underway on the usefulness in treating Lyme?
It is so interesting you asked this question, because I just came from a local conference and it was discussed, and I was making some notes on that one. So I have a new student who wants to, I mentioned in my presentation, who wants to explore additional antimicrobials, and that will be on the list.
Also, many thanks for your research, Dr. Sapi, from others. Are there any contraindications of monolaurin during use for chronic Lyme patients who may also have mold exposure and fungal issues?
That's a very good question. Again, the conference I attended just last week talked about that, and I don't recall any contraindication because it was a mold lecture, and it was some antimicrobial lecture, and they didn't bring it up. But I think a lot of doctors right now looking into this alternative treatment. So I think the data will come out soon.
Thank you. Does your work and research overlap with Dr. Spector's work?
Dr. Spector obviously was one of the pioneers also to look into cancer research and pathogen infection. He believed in Bartonella infection was one of the important in breast cancer. So when we're gonna look at those co-infections in breast cancer tissue, we're definitely gonna look for Bartonella.
And thank you, Dr. Sapi. Very much appreciated for an excellent session. There's one other question. Have you looked at phages therapy to try to penetrate the biofilms?
Yes,
or other DMSO treatment?
I even have a virologist who knows phages, and we started to do now. You have to understand, I'm working with students, and some students are very good, and some students sort of disappears. So we started the phage project, and somehow it wasn't finished, but one of my students actually looking to start again a phage, finding some phage. This colleague of mine, this is what she does. So that would be a perfect collaboration between two of us.
And that's it for questions today. Many thanks in the meeting chat for you and this wonderful session, Dr. Sapi.
Thank you, Athena, and thanks for everybody.
Thank you again, Dr. Sapi, for your presentation. Thank you, Athena, for a wonderful moderation and helping us with reading off all the questions and getting engaged in the audience. And thank you to the audience for your questions. I just wanted to remind you that our Lyme disease presentations and tick-borne disease presentations continue tomorrow. We do have Dr. Susan Cork from the University of Calgary, who will be presenting on the ecology and epidemiology of Lyme disease in Western North America. Her presentation starts at noon Eastern Time on Friday, May the 12th. And then we will obviously continue next week with a series of 4 more presentations throughout the week. Just a reminder to take part in our challenge, wear green, take a photo and share it with us to help spread awareness of Lyme disease and tick-borne diseases. You can even send photos of your creative artwork. So if you like to paint or draw or poetry or even pottery, feel free to take a picture and send it in. All entries received will be entered into our draw, which will be occurring at 1 o'clock. During our last presentation on May 31st, you can send your photos into cluttered@gmail.com along with your name and email, and you'll win a chance of one of 4 $25 Starbucks e-gift cards. And finally, a reminder that TICNET Canada will be having its in-person scientific symposium this fall in Toronto, Ontario, October 24th to 25th, and registration abstract submission should be opening soon. I know the planning committee is trying to get all the logistics together, and we're hoping to open up that registration and abstract submission by June 1st. So stay tuned. Hopefully we'll have some more details. Again, thank you, Dr. Sapi. Thank you, Athena, and the audience. And we hope to catch you tomorrow at Dr. Susan Cork's presentation. Have a great rest of your day and week. Take care, everybody.
Thank you, everybody.
Thank you. Bye-bye.
Bye.